Nivedita is here by taking a video on the topic salient features of human genome. See, the topic is continued. In the previous video, I covered what is HGP and why do we call it as a mega project, and as well as goals and objectives of HGP, that is Human Genome Project. Okay. So this video is in the continuation of the same topic, and this will cover salient features as well as application. Features of human genome. First means how much work is done in this project. So the findings are human genome contains 3164.7 million base pairs. Okay, and an average gene consists of 3000 bases, and the largest gene consists 2.4 million bases. Okay, these are the findings one, and the function of over 50% of the discovered gene are not known. Only less than 2% of genome codes for protein, means still there is a question, what is the role of that or the remaining genes, okay, whose function is not known, then what is their work? Because this is find out that only 2% of the genome codes for protein, then what about other remaining part? So still this is a question, and there are so many of repetitive sequences, and this repetitive sequences throw light on chromosomal structure and evolution, especially the topic evolution. Because if the part which is unknown, means whose function is not known, if there some changes are going on, then this it will not be seen because they are not expressed at all. Okay, and this repetitive sequence means some sequence is there, a particular sequence is there that is repeated, so that is called as repetitive sequences. So it make up a large portion of human genome, and the largest one means the uh, chromosome number 1. It has the 2968 genes, and these are the maximum number of genes present on the chromosome, and the least number of genes are present on the Y chromosome. Okay, that is 231, and scientists have identified about 1.4 million locations where DNA differs in single base only. Okay, means same sequence is there and that is differ by only a single base. Okay, this is the findings, one of the findings. Now, methodologies of HGP. What were the methodologies, okay, used in the HGP? So before we go to the methodology, before that we will go what were the approaches, which approaches were involved. So first there are two important approaches. One is ESTS, that is expressed sequence text. Okay, and this ESTS focused on identifying all genes that expressed as RNA, means the name is suggesting ki how much, means all those genes that is expressed as RNA. Okay, that is the ESTS. And second approach is sequence annotation. Okay, sequence annotation, and that means simply sequence whole set of genome that included all coding and non-coding sequences. Understand? Expressed means केवल वो वाला part ढूंढा जाए वो वाला पोर्शन जीनोम का जो आरएनए के फॉर्म में एक्सप्रेस हो सकता है, एक ये अप्रोच थी। एंड सेकंड अप्रोच वाज कि सारा ही होल सेट ऑफ जीनोम उसको फाइंड आउट करना है, इट इंक्लूड्स कोडिंग एंड नॉन कोडिंग बोथ द सीक्वेंसेस। ओके, तो दीज वर द टू अप्रोचेस, simple thing. So the methodology is, okay, what methodology is there? So steps are, first of all, total DNA from the cell is isolated and converted into segments. Okay, first is isolation and fragmentation. This is the first step in short. Second step is all those fragments, they are cloned, means they are made, means so many copies of those fragments are prepared, and that is called as the cloning. Okay, and this cloning is done using suitable hosts like BAC and the YAC. BAC is bacterial artificial chromosome and YAC is yeast artificial chromosomes. Okay, and then the fragments are sequenced. Okay, first what is, the DNA is isolated, second is fragmentation, third is the cloning, and fourth is the sequencing of the fragments. Okay, then what is done, the sequence were then arranged on the basis of certain overlapping regions. Then specialized computers, computer based programs, these were developed, and the sequence were annotated and assigned. Okay, and the next task was to assign the genetic and physical maps. So this was the methodology used in the HGP. And now what is the applications? Okay, methodology is over. Application is what? The complete sequence. The complete sequence of human genome. It will enable new approaches to the biological research. Means it will help and will give a way or will light, okay, on this researches, and all the genes in a genome or a tissue or an organ or in tumor that can be studied. Okay, all the genes, and this also helps in understanding the chemistry of life. Okay, so these are the important applications. Okay, first it will help in biological researches. Secondly, all the genes of a tumor or enough that can be studied, and it also helps in understanding the chemistry of life.